It's clear there's a lot of excitement around this solution, but there are some other solutions out there that it can be compared to. Can you speak to that a little bit?
Sure. And well, if you look at actual inlays for keratoconus, the first that was used were intracorneal ring segments, CTAKs and Kera rings, and they indeed had some success. There are some issues with biocompatibility, and certainly they were not customizable to the individual patient. You may have heard of the CARES procedure. That's an acronym for corneal allogeneic intracorneal ring segments. So CARES is essentially a tissue mimic of the intracorneal ring segment procedures. CTAK was really developed in an entirely different way because we were looking at doing an intrastromal lamellar keratoplasty procedure to improve corneal topography and vision. And this has, again, evolved to give us extremely good results in our study. Patients improved on average 6 lines of their uncorrected vision and indeed have some kind of a wow effect. The next day, they are seeing a lot better. The other important attributes of CTAK are, number one, we are able to use a custom approach using patients' corneal topography and tomography data. Currently, we're using the Pentacam, but along with CorneaGen, we are trying to expand this to other topography and tomography instruments. We're able to take individual corneal data, translate that into an appropriate custom inlay, which is then prepared by CorneaGen using your standard eye bagging techniques, tissue preservation techniques, to yield a preserved, shelf-stable inlay, which can optimize the patient's visual acuity. Important in this is indeed, in addition to the customized approach and the meticulous preparation of the tissue, we're also able to give the surgeon a suggested plan to optimize placement of the CTAK inlay, as well as its location with regard to the cone and with regard to the pupil. We have found long-term that the CorneaGen inlay tissue is biocompatible, very clear early on, and maintains clarity over a long period of time. In addition, the gamma irradiation makes the tissue very easy to use. It has a degree of stiffness to it that lets the surgeon apply it with fair ease. We think of CTAK as really being a surgical ecosystem. We have meticulously prepared tissue. We have specifications defining the custom tissue, specifications defining placement of the tissue, a surgical line of equipment designed to make this procedure something that's of fair ease for the surgeon and the patient. So the surgeon can embrace this ecosystem again, and I think get excellent patient results very quickly.
No, absolutely. And it's certainly that. I think it's that comprehensive nature that have made so much excitement around it from surgeons, and to the point that our wet labs that we have and our trainings are booked, and now 60 plus surgeons that have done at least one, and dozens in the waiting to continue with this great comprehensive solution.
And we're getting excellent feedback from the surgeons. I think they're very happy with the results, and more importantly, the patients really appreciate the results.
Well, thank you so much for your time today, Peter. Really enjoyed the time together and sharing more about the history and evolution of CTAK. For more information and to integrate CTAK into your practice, please contact us at CTAK@CorneaGen.com. You can also check out our extensive library of great educational content, surgical pearl videos, and recorded webinars at CorneaGen.com. Thank you all very much, and thank you, Peter.
Thank you, Ernie.